Good day, this is Professor Will, CBMD PhD. Today, Saturday, August 24th, 2019. It is 23:29 and 9 seconds Eastern Standard Time. And it's a continuation of my scientific research on pathophysiology, particularly DNA replication. So let me continue here. DNA replication in Escherichia coli is a carefully regulated process involving multiple components representing more than 20 genes participating in duplication of the genome. The process is divided into distinct phases: initiation, elongation, and termination. The synthesis of a new chromosome involves an array of complex protein assemblies, acting in sequential fashion in a carefully regulated and reiterated overall pattern. The scheme for DNA replication is under careful genetic control. The process is localized on the DNA structure by both DNA sequence and topology and requires specific protein DNA and gene products that are required for the initiation of new rounds of DNA replication. The identification of temperature-sensitive DNATs mutants was one requirement for understanding DNA replication. The second requirement was the development of systems that could be biochemically manipulated but that represented all or more of the authentic DNA replication process in E. coli. Several successive systems offered increasing advantages. Systems in which permutable cells allowed free access of small molecules to minimally disturbed chromosomes were the earliest. These systems allowed definition of the energy and cofactor requirements for the elongation phase of DNA replication. The limitation was that they did not permit access by macromolecules to the replication apparatus and therefore did not allow complementation of defects in proteins required for DNA replication. Also, such systems did not allow the initiation of new rounds of replication. The development of lysate replication systems rested on the recognition that the failure to maintain the complex process of DNA replication in early studies was due to dilation of the components, resulting in disassembly of the replication structure and loss of functions required for DNA replication. The concentrated lysate systems depended on the bacteria chromosome, but it was quickly recognized that small bacterial phage chromosomes could be utilized as exogenous templates for DNA replication because they permitted the addition of proteins to allow complementation of defects of DNA replication. Lysates made from mutants defective in a specific step of DNA replication could be used to define the step at which the defect occurred and to identify the protein's product complementing the defect. This allowed assignment of protein products to genes. Such systems, however, did not allow the study of initiation of new rounds of DNA synthesis on the host chromosome. Two theoretical shortcomings of such systems are that such systems might not define all the proteins required for DNA replication by the host, and 2i, that such systems might require a protein for replication of the phased DNA not ordinarily required by the host chromosome. The following general point derived from these studies is worth remembering. Although E. coli contains numerous proteins that have overlapping or similar enzymatic function, the participation of a protein in the replication process is carefully regulated, reflecting a specific role. DNA polymerases are the best example. Each of the three recognized DNA polymerases, E. coli, has similar enzymatic capabilities, but ordinarily only DNA polymerase III catalyzes replication. This restriction of activity can be partially explained on the interactions. DNA replication in E. coli is bidirectional and symmetrical. Development of the field. This article will focus primarily on Escherichia coli as a model organism, with the assumption that what is true for E. coli is generally true for other prokaryotes. In gram-positive bacteria such as Bacillus subtilis, this assumption has been largely substantiated. Sequencing of several prokaryote genomes has revealed homology to many of the genes involved in E. coli DNA replication. The development of our understanding of DNA replication in prokaryotes depends on the combination of biochemical and genetic approaches. The development of our understanding of DNA replication in prokaryotes depends on the combination of biochemical and genetic approaches. Using several selection techniques, many laboratories isolated E. coli mutants that were conditionally defective, usually temperature sensitive, and DNA replication. This method of identifying genes involved in DNA replication assumed that defects of such genes resulted in the death of the cell. When a large series of mutants was assembled, they fell clearly into two broad categories: those in which DNA replication ceased abruptly following a shift to restricted conditions, and those in which DNA replication ceased slowly. The former class is called fast stop and latter slow stop. The first category represents cell-containing mutations and gene products that are required for the elongation phase of DNA replication, and the latter category contains cells with defects and gene products that are required for the initiation of new rounds of DNA replication. Control of DNA replication. Control of DNA replication relies on the regulation of new rounds of replication. In E. coli, there are two components of control: the DNA A protein and the structure of the origin of DNA replication, ORIC. The region of the E. coli origin of DNA replication is at 85 min on the genetic map based on a total of 100 min. On the sequence E. coli genome, the origin is located between nucleotides 3923371 and 3923602. Thus, there is a fixed site on the E. coli genome that represents the appropriate place for the initiation of DNA replication. The DNA initiation is referred to as macro initiation as opposed to repetitive initiation, which must occur multiple times during the elongation phase of DNA replication. The latter is referred to as macro initiation. It seems that the requirements for macro initiation at the ORIC region include those needed for macro initiation plus additional requirements. There is a region of approximately 250 BP that must be present for DNA replication to initiate. Macro initiation. Several features regarding this region are notable. There are multiple binding sites for DNA A protein, the DNA A boxes. This is a 9-nucleotide sequence that has been shown to bind the DNA A protein. There are also multiple promoter elements suggesting the involvement of RNA polymerase in macro initiation. Possible DNA gyrus binding sites are also present. Another notable feature is the presence of multiple DAM methylase sites GATC sequence. The definition of the ORIC region depends on cloning of this region into plasmids, constructed so that replication in the plasmids depends on the function of the ORIC sequence. This has allowed the development of in vitro assay system of macro initiation of DNA replication. The cloning of ORIC confirmed that the specificity of macro initiation in E. coli resides in the origin. The primary protein actor in macro initiation is the DNA A protein. This protein binds at multiple sites within the ORIC structure as noted. In addition to binding at the DNA A box consensus sequence, the DNA A protein displays a DNA-dependent adenosine triphosphate ATPase activity and appears to display cooperative binding properties. This suggests that the possible role in initiation is a change of conformation of DNA and DNA A protein interaction. The DNA A protein also binds in the promoter region of the DNA A gene, suggesting autoregulation, which is supported by genetic study. It appears that the DNA A protein must act positively to initiate DNA replication in E. coli. Both protein and RNA synthesis are required for macro initiation to occur in E. coli. The macro initiation phase may be further subdivided into stages. The earliest step involves the binding of DNA A protein to the ORIC structure. This results in the conformational change of the origin. This complex then binds the DNA A, DNA B, and DNA C protein. They are also required for the elongation phase of DNA replication. DNA C plays a unique role in the delivery of the DNA B protein to the replication structure. The resulting complex appears to unwind the DNA strand since the DNA B protein functions as a helicase and ATP-dependent unwinding activity. This allows the binding of single-strand binding SSB protein, which allows priming such as that which occurs in the elongation of DNA replication. This stage is followed by the propagation of micro-initiation and elongation phases of replication. Thus, the proteins required for the macro-initiation of E. coli DNA replication appear to include DNA A and DNA C, which have specific roles, as well as DNA B, SSB protein, JARS, the DNA G primase protein, and the replicative apparatus of DNA polymerase. 3. Halo enzyme complex. Studies also suggest a direct role of RNA polymerase and the macro initiation of DNA replication. Good day, this is Professor Will, CBMD PhD. Today, Sunday, August 25, 2019. It is 00:40, 10 seconds Eastern Standard Time. And this concludes my reading on pathophysiology, particularly DNA replication. Hope you enjoyed listening. Have an excellent day.